Welcome to Chapter 13 of Microbiology. This chapter discusses the relationships that humans and microbes create, the relationships that are beneficial for us, as well as relationships that can lead to infection. During the birthing process, a baby is colonized by a mother's microbiota. Homework 1 discussed some of the beneficial features of this new colonization. For example, the bacteria that happens to make glycoside hydrolase. Glycoside hydrolase allows the baby to digest the mother's milk. In the absence of glycoside hydrolase, we would not be able to feed our young. Additionally, the colonization of a baby with normal microbial flora is that first line of immune defense from pathogens. So by having normal flora taking up space, babies then are able to be protected from unnecessary pathogens that they may encounter. So the major factors in the development of an infection first begin with the portals of entry. And so I'd like you to think for a moment about ways that a microbe could enter your body inadvertently. So some of the ways that a microbe could enter your body are through the oral cavity, nasal cavity, urogenital tract, GI tract, lungs, so respiratory cavity. And so these mechanisms of entry do control the sites of infection. So the various portals of entry are often protected by immune barriers, and we'll discuss the immune barriers a bit more in chapter 14 and 15. So for microbes to be able to infect, they first must be able to attach to your host cells. And so attachment is critical. Reflect back on a few of the mechanisms of attachment that you have learned in chapters 4 and potentially 7. So microbes attach by using fembrae, for example. So the fembrae is the hair-like bristles on the surface of the bacteria. Fembrae are necessary for attachment, and if this microbe cannot attach, such as E. coli in the gastrointestinal tract, if you have a pathogenic version of E. coli, if this pathogenic version cannot attach to our microvilli in the intestinal wall, then there will be no infection. The normal peristalsis that takes place, the contraction in the intestine, will instead flush the bacteria out. And so there are things that can increase our susceptibility to microbes, such as weakened immune defense. And so I want to draw your attention to the fact that both elderly and young are possessing a weakened immune system. So upon birth, babies have not been exposed to all of the microbial flora, the environmental cues, so they have not had their immune system educated against these potential pathogens. And then again, they have not had their immune system educated against what is normal. So as a consequence, there's a lot of growth and development in a young immune system. And then as we age, our immune system begins to contract. And we find that the immune system of an elderly adult actually starts to resemble that of a young person because of the contraction and loss of those immune cells that were developed in the bone marrow. In some cases, there are genetic defects that can cause issues with immunity. So if you are lacking a gene that is necessary for an immune cell, secretion of mucus that could protect the lungs or things like that. The procedures such as surgery and organ transplants, these are going to contribute to the likelihood of disease. And we often consider these and organic diseases comorbidities. So if you have other issues going wrong with your patient, they're more likely to be susceptible to an infection. And then, of course, we don't really consider it as often as we should, but physical and mental stress. So if you are working a lot and you have a really high course load or you're stressed out, you tend to have a much more deficient immune system than that of someone who has a well-balanced life. And again, other infections are going to contribute the likelihood of a secondary infection taking place due to kind of your immune system fighting on two fronts. So the establishment of an infection relies on those portals of entry. And so portals of entry can deal with both exogenous or endogenous sources. Exogenous, exo meaning from outside of the body. So exogenous sources are when we pick up a free-living microbe or we pick up a microbe from contaminated food sources such as, infective Escherichia coli. Endogenous sources, however, are sources of microbes that are already on the body or normal flora. And so Clostridium difficile, for example, is already in the GI tract. And so Clostridium difficile, when we take antibiotics, if all the microbial flora from a gastrointestinal tract is wiped out, we then can have an endogenous infection with Clostridium difficile. So the portals of entry that we will discuss, of course, skin. So our skin is a first line of defense. It is a continuous barrier, but when we have nicks, abrasions, and things like that, we now have an opportunity for other microbes to get in, which is why we often, if we have a cut or a scratch, we immediately flush that tissue out. We, treat it and cover it with a Band-Aid or necessary ointments if we feel that it is at bacterial risk. Gastrointestinal tract. If you've ever traveled, we are often very cautious of food, drink, and other materials from less domestic countries because they have a very different sanitation system than that of which we experience in the U.S. So as a consequence of that, there's more likely an opportunity for fecal material, fecal contaminants to be within those water supplies. And so gastrointestinal and tract infections occur from contaminated foods or drinks, often with enteric bacteria. We can also have portals of entry through the respiratory tract. So we're currently in flu season, but of course, yearly we have issues where there are respiratory infections that pass from, person to person. These are going to be transmitted through oral droplets or by direct contact with contaminated sources. There's the urogenital tract. So more often than not, these are sexually in orientation. So here's a listing of a few of the sexually transmitted diseases. And then, of course, transplacental. And again, usually these sexually transmitted infections moving from person to person can then also go from mother to child in a transplacental route. And so pathogens that infect patients during pregnancy are represented by the acronym STORCH. And that begins syphilis, toxoplasmosis, other diseases which are hepatitis B, AIDS, or chlamydia, rubella, cytomegalovirus, and herpes simplex virus. These are common infections that happen to manifest during pregnancy and are of greater concern to that of a pregnant mother and their child. For us to have an infection, you must reach the minimum infectious dose. And so first, microbes have to be able to attach to cause infection. Second, they have to reach the infectious dose. The infectious dose varies depending on the type of microbe. The infectious dose, for example, of measles is as little as 1 virus, which is really, really low, versus you have things such as the plague, which is 100 to 500 cells. And so we would consider the measles virus much more infectious than that of the bubonic plague. And so any type of microbe that does not reach the infectious dose will not cause infection. And so to reach the infectious dose, the microbe must first be able to attach to your respiratory cells or your host cells, depending on the infective location. In the case of measles, it would be your respiratory tract. And upon attaching, then it has to colonize that area and begin replicating to reach the infective dose. So mechanisms of attachment include fembrae, so the proteinaceous fibers on the surface of a bacteria. They include flagella. So flagella is not solely in use for movement. It is also used to attach to host cells. Components of the glycocalyx, so the slime layer or the capsule, aid in attachment to the specific host cells and enable the microbe to enter. Cilia or suckers, so suckers and hooks and barbs, these are all part of Helminth mechanisms of attachment. And then of course shown here, there's also viral spikes. And so viral spikes are proteins on the surface of a virus. And those proteins are highly specific for proteins expressed on the surface of a host cell. And they are necessary for attachment. So here's just a few lists of adhesion properties that are some of the bacteria that we'll be discussing and some of the viruses that we'll cover throughout the semester. And so Escherichia coli, which causes diarrhea, has a lot of fembray attachment, so it must be able to attach and persist within our gastrointestinal tract. See the same thing for Shigella and Salmonella, both of which cause gastroenteritis. And then poliovirus or AIDS, both of these are going to have distinct receptors on their host cell surfaces that they're specific for to enter and attach. So to be able to survive and cause infection by reaching an infectious dose, these microbes have to be able to survive that of the immune system. And so one of the first initial responses that are going to take place is the second line of defense formed by that of phagocytes. So phagocytes are eater cells, and phagocytes are going to be necessary to clear a lot of our bacterial infections that will try to colonize on our host cell surfaces. And so as a consequence of this, these microbes have developed antiphagocytic factors. And so an example of an antiphagocytic factor would be that of a capsule. A capsule can be used to avoid phagocytosis. Then, of course, there's distinct microbes or bacteria such as Staphylococcus or Streptococcus that produce a virulence factor called leukocidins. Leukocidins are going to be very toxic to white blood cells, and so if the white blood cell comes in close association with Staphylococcus or Streptococcus bacteria, the release of leukocidins will cause the apoptosis of these white blood cells. The slime layer or capsule, which are components of the glycocalyx, make phagocytosis more difficult, and so the resistance to phagocytosis, or the ability to survive after phagocytosis, is a distinctive virulence factor that allow distinct microbes to survive. An excellent example of this that we'll discuss this semester will be Listeria monocytogenes. So first, the microbe must enter into the host cells. So this is a descriptive example of Salmonella. So this is Salmonella and the secretory system that is necessary to digest the contacts between cell-to-cell interactions. And so it's going to actually cause the ruffling. It's going to get pulled in through these cells through a vacuole. And then if it can actually transcytose through the membrane of the host cell endothelium, it's then going to move into the vascular system and cause a systemic infection. So systemic infections are of a much greater concern for the body, because now you're dealing not only in a localized infection, but is moved to a new site of location. So the virulence factors that allow the microbe to do these processes are that of exoenzymes. So the exoenzymes were described in the previous slide by digesting cell-to-cell contacts. So exoenzymes of, for example, Salmonella are going to allow them to enter in through the intestinal tract, moving these cell-to-cell contacts and allowing the microbe to get into the systemic blood supply. And then, of course, some microbes, especially those that are going to cause severe GI distress, are going to have toxigenicity, meaning they're able to secrete toxins at the site of multiplication. And so toxigenicity, such as toxigenicity secreted by Shigella secretes Shiga toxin. And so Shiga toxin will cause systemic damage within that site of infection, result in severe inflammation and things like that. And so there's two distinct types of bacterial toxins. There's an endotoxin and an exotoxin. So endotoxins are only secreted after the bacterial cell is damaged. So it is a toxin that is a normal component of the microbial surface. And so in the case of gram-negative bacteria, that endotoxin is lipopolysaccharide. And so that endotoxin is lipopolysaccharide, and it's a part of the gram-negative cell wall. It's that major outer layer. And so when the bacterial cell is lysed, the lipopolysaccharide components are released, and then they can act within that site of infection upon where the microbe was burst. Exotoxins, however, do not require the microbial cell to be lysed. Instead, the bacterial cell will secrete this distinct toxin. And so an excellent example of an exotoxin is a hemolysin. A hemolysin is necessary to digest blood cells. So Streptococcus pyogenes was an excellent example of a bacteria that secretes a hemolysin. And so hemolysins are highly specific. They have both an active component and a binding component. And so the A and B toxins work in a concerted effort to cause damage to the host cell. And so here's two differences between endotoxins and exotoxins. So exotoxins are highly specific. They can target distinct organs, they can target the muscles, blood, and they can cause distinct dysfunctions within the site of infection. Endotoxins, however, because they are acting at the site of bacterial lysis, they're going to cause general physiological effects such as fever, aches, and can cause septic shock with complete dissemination. So here is a brief compare and contrast of exotoxins and endotoxins. And so toxicity for exotoxins, they're toxic in tiny amounts because they're highly specific. Endotoxins, however, are only toxic at higher doses, so a distinct quantity of lipopolysaccharide being lysed from bacterial cells can result in eventual shock because of the higher dosing. And so the A and B exotoxin, when they go through the function of action, the A component, right? So the A component must associate with the B binding component. So I consider A component for active, B for binding. The binding component is actually what brings the active component to the host cell membrane. So without the B component, you would not have the association of the A component with the host cell membrane. And so once the B binding component is attached to the host cell membrane, it's going to enter in through endocytosis. Once the A and B exotoxin has entered in through endocytosis, the active component will dissociate due to a pH change, and then will move off and act on the ribosome. The B component, however, then is going to be shuttled back out and can associate with other active components and interact with the same cell types.
AB toxins are proteins that consist of two parts, A and B. Portion is an enzyme that constitutes the toxic part. The B portion binds to host cell receptors. There are several mechanisms by which AB toxins enter the cell. One mechanism involves the uptake of the toxin after binding by endocytosis. After endocytosis, the contents of the vacuole become acidic, causing the A and B portions to separate. The A portion enters the cytoplasm of the cell and exerts its toxic effect, while the B portion is removed from the cell by exocytosis. Different microorganisms produce A toxins with different activities. The A toxin produced by Corynebacterium diphtheriae carries out the transfer of ADP ribose to elongation factor 2, EF2, and thereby inhibits protein synthesis. ADP ribosylation is a common action of AB toxins.
So that is just one example of AB toxins. We were talking about hemolysins. And so hemolysins, if this was an AB component with hemolysins, what it would do is cause the lysis of a red blood cell. So the processes of infection and disease actually mirror that of the bacterial growth chart. So you'll see that the bacterial growth chart, if we were to overlay it, would have a similar effect. And so that's not surprising, because as we think about the symptoms chart is actually related to that of the bacterial growth chart, simply because our symptoms are highly correlated to the quantity of microbe that we have. And so the incubation period is the time from initial contact. So the incubation period is correlating with that of the bacterial growth chart, or the lag period, because there's a low quantity of microbial growth. So incubation period, we have very few symptoms. You just kind of feel a little off, and so you don't actually know what's going on at this stage. The prodromal period, however, you have much more vague feelings of discomfort, and so this is when you are starting to notice that you're actually sick, but you don't have a distinctive symptom. You just don't feel very well. Now, once you have proceeded to the period of invasion, the period of invasion is this entire area. And so the period of invasion is when you have distinctive symptoms. So if you have a gastrointestinal infection, you are now having stomach aches, vomiting, diarrhea, whatever is coordinating to that type of infection. then eventually your microbial load will reach the height of infection. So this, when we compare it to the bacterial growth chart, this would correlate to the exponential growth period. So this is when the microbe is growing, and then eventually it reaches its height before going into the stationary period. And so the period of invasion consists of the logarithmic period, right? So exponential growth, as well as stationary period. And so after we reach the stationary period where the rate of microbial growth is the same as the microbial death, we then move into a convalescent period. This is when your patient is getting better. And so your patient now is in the convalescent period, which corresponds to the death phase of the microbial growth chart. And so because the microbial load is going down, your patient then is starting to feel better, the symptoms are declining, and eventually they're going to remove all quantities of microbe from the body. So patterns of infection consist of localized or systemic infections. So localized infections could be like a boil. They could be a localized infection where you've got possibly a wound on your foot or your hand. They're going to stay at that site of infection, and they're not going to move from that site of infection. However, you can have systemic infections such as influenza virus, so the flu. And so it begins as a respiratory infection, but often because it infects the vasculature upon that, it's also often systemic, which is you have disseminated periods of malaise and overall symptoms of fatigue. And so focal infections differ from that of a systemic infection in the sense that the focal infection is when you have a localized infection, but then that infection breaks off. And so a good example of this would be dental issues. So dental issues form severe plaques and can cause cardiac issues because the plaques in your mouth can break off and then reach your vasculature and cause damage to your heart. So that would be an excellent example of a focal infection, because the focal infection was actually consisted and localized in your mouth, but then it was manifested in other tissues because the plaque broke loose and moved to a new location. Infections can be described as being mixed. So often we have infections that are not one microbe only. And so mixed infections are polymicrobial. So you have multiple microbes that will grow at the site of infection. You can think of a mixed infection as that of, say, for example, a biofilm type infection, because you've got an orchestrated effort between microbes to cause disease. The primary infection, when we think of a primary infection, is the first time you have that initial infection. So an excellent example of that would be a UTI. So you can have a primary UTI, right? But that primary UTI can actually spread and become that of a kidney infection, or in this example here, it's talking about a secondary bacterial infection, a secondary infection in the vaginal tract. And so that secondary infection in the vaginal tract could have been a manifestation of antibiotic use causing a yeast infection, or it could be a secondary bacterial infection in the vaginal tract. By this image, it's showing distinct yeast cells. So patterns of infection can be acute or they can be chronic. And so when we think about these patterns of infection, the common cold is an excellent example of an acute infection because it begins within 2 to 3 days after contact, but then it's completely resolved after about 10 days, and it can be resolved without therapy. Versus chronic infections are when you have an infection that can persist over a very long period of time, or potentially even a lifetime. These sorts of infections have periods of latency and reemergence. An excellent example of that is cold sores. Cold sores are caused by herpes simplex virus 1, type 1. And so herpes simplex virus 1 can cause reemergence when a patient is stressed or having other issues. They often have a flare-up of cold sores. The signs and symptoms of inflammation consist of fever, pain, soreness, and swelling. And so this is how we know we have some sort of illness taking place. And so, for example, when we think of edema, right? Edema is the accumulation of fluid. So think about why we would have fluid accumulate at a site of infection. Our immune system has to move cells to the site of infection to remove the infectious agent. So as a consequence of that movement of cells, you have an accumulation of fluid in the site of infection and edema because you have a movement of immune cells to try to fight off that infection. Granulomas and abscesses are phagocytes that will actually wall off the microbe, preventing the spread of the infection if it can't be cleared by normal processes. And so lymphadenitis is one of the key mechanisms that physicians use to identify a patient who has an infection. And so they will palpitate around the throat. And so typically if you have an upper respiratory infection, you will have then lymphadenitis around the throat, showing that the lymph nodes are draining the upper respiratory tract. So signs of infection in the blood, you can actually diagnose patients by looking at the blood count or doing a differential complete blood count. Leukocytosis is an increase in white blood cells, so presence of white blood cells in urine or presence of excess quantities of white blood cells in the blood suggests that the patient is undergoing an active infection. You can also have leukopenia, so penia meaning a reduction in white blood cells. So patients who suffer from HIV, they move into the category of AIDS when their CD4 T cell count moves below 200. Patients also may suffer from septicemia if microbes reach high quantities and numbers in the blood. And so we can count the number of microorganisms in the blood. And so if they're present there in high numbers and they're actively multiplying, then that means that your patient is septicemic. However, if the patient has small numbers of bacteria in the blood and they are not necessarily multiplying, this is only considered bacteremia. So septicemia means that the microorganisms are actively multiplying and they're present in large numbers. Bacteremia means that they're small numbers, but they may not be multiplying. So it's a much less severe disease than that of septicemia. Likewise, viremia is a small number of virus particles present in the blood, but may not be multiplying. So viremia is much less severe than a virally caused septicemic infection. Patients who are asymptomatic often go completely unnoticed because we don't go into the doctor for no symptoms. And so as a consequence of that, patients who are asymptomatic can still be spreading infection depending on the microbe type. Portals of exit. So when we think about the way microbes get in, they also leave by similar mechanisms. And so respiratory tract, our coughing, can actually spread microbes at a distance of about 10 feet, depending on the type of microbe. But we can also spread disease due to nasal drainage, so blowing our nose, not washing our hands, spread of saliva. Our skin scale, so if we have microbes or open lesions on the surface of our skin, then we can spread the microbes from the skin route. Of course, fecal exit, so dissemination of feces and contaminated feces coming in contact with other persons can cause pretty severe infections. Urogenital tract, so contact with infected urine. So these are types of infections that would probably be disseminated with a healthcare practitioner, so nosocomial infections. And then, of course, removal of blood. So coming in contact with blood can be highly infectious and contribute to the dissemination of infections such as HIV from patient to healthcare professional. Persistent infections in microbes. So as I'd mentioned before, persistent infections consist of periods of latency and reemergence. And so patients who are experiencing a latent infection will have periodic periods where they're active and producing recurrent disease. And then, of course, depending on the type of infection, the patient may or may not shed the virus during the latent stage or shed the microbe during latent stage. So there are some infections that, such as Ebola, where if you were in a latent stage or you are not showing signs or symptoms, you are actually not infective. However, there are distinct STIs that the patient, although they're not showing genital warts, for example, they may still be infectious during that latent stage. Chronic carriers. Chronic carriers are patients who have a latent infection and they continue to shed the infectious agent. So a chronic carrier is a person who is always infectious. And due to infections, we often have long-term or permanent damage. So an example is Streptococcus pyogenes. Streptococcus pyogenes is at a great risk for a long-term heart or endocarditis as a sequelae. The sources and transmission of disease consist of both the reservoir and the distinct source. And so a reservoir is the natural habitat of a microbe. So excellent example of this is influenza virus. The reservoir is actually in the gastrointestinal tract of birds because birds can harbor the virus without showing signs or symptoms. So this is a location at which the virus can reach high microbial loads without causing damage. Other reservoirs can be, for example, humans. Diphtheria, we are the natural reservoir for this bacteria. There are certain bacteria that have animal carriers, so they persist within the animal without causing disease. An excellent example of this would be tularemia, which is often disseminated by rabbits. Soil, so the bacillus bacteria that we'll be discussing this semester often will propagate within the soil, so the persistence of endospores in the soil can cause the dissemination of this microbe, so it can reach high quantities of endospores in the soil, persist for long periods of time, and then if we come in contact with contaminated soil, then we can have infection. Water sources. So water sources are often a primary reservoir for gastrointestinal diseases. So Escherichia coli, for example, can persist in water sources and feces and cause severe disease at a later date. Sources of infection are where the microbe are actually acquired. So for example, you could acquire the infection from the source, such as soil, which is also a potential reservoir. So for example, if you have a housemate who wears shoes outside, they pick up endospores on their boot, they wear their shoes in the carpet, then you happen to come in contact with the carpet, or then you pick up the endospore from the carpet that was from actually a secondary source. And so the source is the individual or object from which the infection is actually acquired. It does not have to be the reservoir, but it could be the reservoir. Excellent example is someone contaminates or doesn't wash their hands. They touch a doorknob, you pick the microbe up from the doorknob. Could be that of a roommate. So the roommate happens to have an infection and you pick up the infection, or possibly you pick it up from a child, your daughter, family friend, or something like that. So living reservoirs. So living reservoirs are considered various carriers. And so when we think of a carrier, this is a person who is going to spread the disease from themselves to another person. Carriers do not have to be infected with the agent. There are asymptomatic carriers, so this is a patient who has no symptoms but is infected with the disease, but they're in that early stage, the asymptomatic stage, or the incubation period stage. The passive carrier, however, this is a person who is contaminated with the microbe, so it could be on their jacket, that could be on their sleeve, stethoscope, and they carry this microbe unknowingly from person to person, or they carry it in the form of a fomite. So a fomite is an object that is contaminated with the microbial source, such as ties, mops, blankets, clothing, stethoscopes. Excellent examples in the clinic are those of your watch, right? So watches make it very difficult to appropriately scrub up your arms and remove all microbes. And so fomites such as watches, jewelry that you may wear during the clinic. If you like the really cute jackets that they wear, the jackets are great and they probably keep you warm, but those sleeves then can become fomites because the sleeves are probably not being properly washed as you move from patient to patient. So types of asymptomatic carriers include incubation carriers. So incubation carriers are those who are still in the incubation stage of infection. Convalescent carriers are those who are in the convalescent stage of infection. They're recuperating. They're not showing signs or symptoms, but they're still actually infectious. And then, of course, there are chronic carriers. So chronic carriers, as we mentioned before, have periods of latency and reemergence. And so as a consequence of those periods of latency and reemergence, there are times when they don't have symptoms, but they are actually still infectious. So if a nurse transfers a pathogen between a patient without becoming infected herself, the nurse has acted as a chronic carrier, convalescent carrier, incubation carrier, asymptomatic carrier, or passive carrier. Best answer is passive carrier. So the acquisition and transmission of infective agents takes place because most of these infectious agents are considered communicable diseases. Communicable diseases are considered contagious, being able to be transmitted from person to person or from host to new host. There, however, are non-communicable diseases that cannot be transferred from person to person, but instead must be obtained from the natural or non-living reservoir. So an example of this is, for example, a compromised person, right? So immunocompromised. It's when they are invaded by his or her own normal microbial flora. So this example is that of an opportunistic infection that happens within the host. Patterns of transmission. They can be either direct or indirect. So direct contact are when you're actually coming in contact with the infected person, or they're fine aerosolized droplets. So you can see here the aerosolized droplets descending, and depending on the infection, in some cases the aerosolized droplets can actually infect at a 10-foot distance. Indirect contact, these are passed from infected host to an intermediate source. A vehicle can be inanimate materials, food, water, or biological sources. That of fomites, so fomites, as a refresher, could be your stethoscope, your watch, your coat, mops that janitors use to clean in the clinic. It could be the bed that your patient is resting upon, or a blanket that may not have been cleaned properly during laundry day. And so here is a breakdown of direct sources. So direct sources, often STIs are transmitted by direct sources. They don't thrive, they're very vulnerable and don't thrive outside of the body for very long. And so Epstein-Barr virus can be transmitted from person to person, aka the kissing disease. Or, for example, the STI of gonorrhea due to sexual contact. And then transmission of various colds or chickenpox can take place through respiratory droplets. Vertical transmission is from mother to child, so things such as HIV or syphilis can be transmitted from mother to child. There are biological vectors, so West Nile virus is transmitted from mosquitoes. So West Nile virus, or for example, malaria from distinct mosquitoes. Indirect mechanisms of transport are fomites. So fomites where you come in contact with a contaminated source or doorknob. Fecal-oral contact. So a lot of GI infections are due to contaminated food sources, such as Salmonella and E. coli. So this is a person making food, possibly this is ground beef that's not cooked to a proper temperature and now is harboring E. coli. Then you have, of course, direct sources, so aerosolized droplets of fecal contaminants. Things like tuberculosis can be transmitted from person to person in aerosolized droplets and things like that. So nosocomial infections. Nosocomial infections are infections that are acquired in a hospital stay. There's actually new legislation that is moving toward no longer paying for nosocomial infections. So in the clinic, if you are responsible for contaminating your patient due to poor procedures, improper PPE or personal protective equipment, then there's actually a movement to stop paying for those cases because they're growing in number. A big part of that is the way that our healthcare system is currently designed. As a clinician, if you move into a hospital arena, you may have, as a nurse, 6 to 10 patients, depending on the type of floor that you're working on. If you're expected to see those patients 5 times within a shift, and your shift lasts 10 hours, well, that's actually a really low frequency of interaction. And you're due to wash your hands before you go into the hospital room. You're due to wash your hands after you go into the hospital room. So for just those 5 patients, each 5 patients, you're washing your hands 10 times in one visit. So if you visit those patients once an hour every 10, for a 10-hour shift, right? So then you have now washed your hands 100 times in a single shift. And that doesn't count if you did anything abnormal or if you had to give someone a bath, different steps or different interactions. And so the frequency at which we are expected to maintain cleanliness actually causes nurses, staff, and clinicians to reduce the efficacy of their washing. So they wash for shorter periods of time. They don't wash efficiently, which then contributes to increased risks of nosocomial infections, as you can see here. The frequency of cases. So now we're into the epidemiology section of this lecture. So prevalence. Prevalence is the number of existing cases with respect to the entire population, and it's typically a percentage. So if we had an outbreak in class and you had 5 people who were sick, so that would be 5. If the class size was 30, it would be 5 divided by 30 to provide you the percentage or the percent prevalence within that class's outbreak. Incidence, however, is the distinct number of new cases over a certain period of time. So incidence, when you answer questions like this for your exam, you should describe incidences. There were 200 cases of influenza since March, right? And so this is often compared to the healthy public, and so the population is considered within those incidents if you do an incidence rate. Mortality rate. Mortality rate is the total number of deaths. Don't get mortality rate with morbidity confused. So morbidity are how many people are sick or afflicted with the disease. Mortality are how many who have died. So here are some examples of how we represent epidemiological data. So shown here is the number of persons, and it's the number of persons over a period of time. So this is an example of incidence. Then over here it says the estimated numbers of adults and adolescents who are suffering from AIDS, right? And so this has actually been broken down over here into numbers per rates of the population. So this is incidence as well. Likewise here, we now are looking at percentages. So percentages are prevalent. So these are the number of cases of TB since 2008. Right, and so those percentages represent prevalence of disease within each ethnic group. And then over here, these are the cases, right, of TB, and they're broken down based on age, and they are the cases in the distinct year of 2008 relative to the entire population. So this is another incidence report. The types of infections and how we describe those infections. Endemic is when you have infections that are within a localized site. They often have a relatively steady frequency over a long period of time. Excellent examples of endemic infection are that of a food outbreak, because the food outbreak will be distinctive to the number of contaminated food products that were delivered to those locales. Sporadic infections, however, have a very spontaneous, irregular frequency. And so sporadic infections are those that have communicable diseases, but then they're not disseminated at high frequencies, so that they're interacting at different rates over a period of time. So an example of sporadic occurrence is that of measles. So measles is highly infectious, and so contamination with one single virus can cause disease. And so this is when you actually have distinct pods of infection over a period of time. Epidemics. Epidemics occur when you have a number of outbreaks that take place that are higher than expected. So the CDC actually has a website, so you can look at the number of outbreaks of illness and things like that. And we often meet or exceed epidemic thresholds during even a seasonal flu outbreak. And so there's periods of time that influenza will reach and exceed that of the epidemic threshold, and then of course it'll drop back down. And even in a normal regular flu season, the fluctuation of disease often will exceed epidemic thresholds and then go below epidemic thresholds. We get really concerned when there's a pandemic. So pandemic is when we are going transcontinental. We had a pandemic outbreak just a bit ago with the Ebola virus. So Ebola had spread pretty extensively here in sub-Saharan Africa and Liberia. But Ebola then did disseminate to the United States. It disseminated to the United Kingdom, to France, and other locales throughout the world, causing pretty severe disease. So just a quick reminder, Koch's postulates. Koch's postulates were discussed earlier in chapter 4, I believe. And so Koch's postulates, just to remind you of that, you have to make sure that Koch's postulates, you always have the distinct microbe in every case. So the microbe must be present in all cases of disease. If it's not present in all cases of disease, then that is not the microbe that is causing that disease. Once you find that it is present in all cases of disease, you have to then isolate that microbe. You have to be able to grow it in a lab. You have to characterize it, make sure that it is indeed a distinct and pure microbe. And then once you've done that, that microbe then should be inoculated into a new host. And so that new host must be able to exhibit the exact same signs and symptoms as the original host that it was isolated from at the beginning. And then upon that same set of symptoms being observed, then you have to re-isolate that microbe, right? Take that microbe from that secondary host. You have to grow it again in a pure culture, verify that it is the same microbe that was isolated and grown here, and that indeed it is the microbe that was causing cases and the same disease in all cases. So Lyme disease, if it occurs mainly in areas where there are ticks, how would you describe this sort of disease? So it's only taking place where a certain species of ticks would live. It would be considered an endemic outbreak, because an epidemic outbreak would be higher than normal thresholds. Sporadic would be all across the nation, would not have any type of association with a distinct species of tick. Pandemic, it's not describing a global outbreak, so that's not a great choice. The best choice is that of endemic, because the species of tick are going to live within a distinct area, and you're going to have cases that would probably occur over a steady period of time of people who happen to be contaminated with this infective agent due to the tick bite.